Hi, in this video, we're going to talk about lactic acid fermentation, which is one of the anaerobic fate of pyruvate. So, we have already seen that glucose can be converted to pyruvate by a 10-step enzymatic process known as glycolysis. This pyruvate would eventually get into the Krebs cycle, and the products such as NADH and FADH would work like an electron donor in the electron transport chain. And ultimately, it would build up a proton gradient which would be utilized to generate the energy currency of the cell. That means the ATP. Glycolysis itself can generate ATP as well. So this thing is the aerobic fate of glycolysis, because when oxygen is present, the electron transport chain can go on because the terminal electron acceptor is oxygen. Now imagine a situation where oxygen is not present. We'll come to a conclusion why oxygen might be low or why there is a hypoxic condition. We will take real life example. But for now, let's imagine that oxygen is low. In that situation, the ETC and the Krebs cycle might not be very efficient way to generate ATP because the terminal electron acceptor is not present. In that case, what happens is pyruvate undergoes an anaerobic fate, which is the fermentation. It can generate ethanol or it can generate lactic acid, and byproduct of this process refuel the glycolysis pathway, and as a result glycolysis becomes the sole pathway for generating energy for the cell. So let's try to put this reaction into context. Okay, but before that I want to tell you why this kind of lactate fermentation is important. So we know glucose in a 10-step process give rise to pyruvate, and that's the outcome of glycolysis, that's the product of glycolysis, right? But imagine in this particular pathway NAD need to be converted to NADH. So the glycolysis pathway need steady state supply of NAD. If NAD is not supplied, then after a while glycolysis pathway would be inhibited or it would shut down. So there should be a way by which NAD can be replenished under hypoxic or low oxygen condition. So pyruvate is converted to lactate, and this process is a reduction process. Okay, now coupled with this reduction process, which is catalyzed by lactate dehydrogenase, something is oxidized, and that is NADH. NADH, in absence of oxygen, get oxidized to NAD with the help of lactate dehydrogenase. As a result, NAD is constantly supplied to the glycolysis pathway, and glycolysis pathway can go on and produce ATP. This is the sole goal of the fermentation process. The sole goal of fermentation, lactate fermentation process, is not to generate lactate, because eventually lactate would be converted to pyruvate. But the sole goal for fermentation of lactate is to generate or regenerate NAD+. And this is very important. And the key enzyme that helps in this process is lactate dehydrogenase. So let us talk about lactate dehydrogenase in a bit. So the dehydrogenase name suggests dehydrogenation is taking place. Dehydrogenation from what? Dehydrogenation of NADH. So when NADH gets dehydrogenated or otherwise oxidized, it forms NAD+, which can refuel the glycolysis. Now pyruvate is converted to lactate. See the hydrogen in pink. So the ketone group is now converted to a secondary alcohol group. So it's a reduction. So pyruvate gets reduced to lactate, whereas NADH gets oxidized to NAD. These processes are coupled and triggered by lactate dehydrogenase. Point to be noted that lactate dehydrogenase catalyzed reaction is highly exergonic, and the delta G is highly negative. The overall equilibrium of this reaction is strongly favored in the forward direction. Now let us put these concepts into a context, a physiological context. let's say we are performing powerlifting, or let's say resistance training in the gym, or let's say we are sprinting. So in very less time, we need a hell lot of energy in our muscles. Yeah, we have glucose, but that glucose is not sufficient. Even after that, there would be glucose coming from glycogen breakdown, but the muscle would create too much of lactate because our blood cannot efficiently supply oxygen in that short span of time in our muscles. So there would be an anaerobic situation in the muscle. In that situation, ETC would not be very useful to give rise energy. In this situation, glycolysis is the important step. So let's see what happens in the muscle and the liver during this anaerobic state or hypoxic state. So obviously in the muscle, glucose needs to convert it to pyruvate, that's the glycolysis pathway, and in this process ATP would be generated which would fuel the muscle for contraction. Now at the side product, NAD is reduced to NADH as well. Pyruvate would eventually get converted to lactate in the muscle, which would refuel the NAD demand for the glycolysis pathway to occur in the muscle. Now lactate need to be converted again. So how that is triggered? So lactate is transported to the liver. Now in the liver, where the lactate concentration is very high, lactate would be converted to pyruvate with the same enzyme, and pyruvate would then get converted to glucose with the help of the process known as neoglucogenesis. So neoglucogenesis triggering enzymes are already present in liver, so pyruvate can easily get converted to glucose. Now this glucose could be resupplied to the muscle and other tissues of the body. So this particular cycle is known as the Cori cycle. So in Cori cycle, what we see, lactate is generated in the muscle, which gets converted to glucose again in the liver and resupplied to the muscle. So overall in this video, we looked at lactate fermentation and the consequence and the physiological importance of the lactate fermentation, and we also discussed the Cori cycle in that context. So I hope you enjoyed this video. If you like this video, give it a quick thumbs up and don't forget to like, share and subscribe. Thank you.